A diagnosis of RUNX1 FPD means that an individual has a harmful mutation or error in the DNA that codes for the gene called RUNX1. RUNX1 FPD individuals have a germline or hereditary mutation in RUNX1, which means that every single cell in one's body, the cells in your skin, your heart, your bones, your liver, every tissue, even your blood, carry the same RUNX1 mutation. There is a push by the medical community to use the word variant instead of mutation. Since mutation is still most commonly used today, we will use that term in this video series. As humans, we have 2 copies of most genes in every cell. Each gene copy by itself is called an allele. People with RUNX1 FPD have 1 normal RUNX1 allele and 1 mutated RUNX1 allele in each cell of their body. The RUNX1 gene represents the blueprint for the RUNX1 protein. Proteins are the workhorse molecules of life and are necessary for each and every cell, important for all living organisms. In people with RUNX1 FPD, 1 RUNX1 allele is normal. There are no errors in DNA code, and it therefore can produce normal RUNX1 protein. But 1 RUNX1 allele is mutated, and the mutated allele, depending on the mutation type, may produce an abnormal RUNX1 protein or no protein at all. Today there are over 100 different germline RUNX1 mutations that cause RUNX1 FPD. In general, people with RUNX1 FPD have half the normal levels of RUNX1 protein. The RUNX1 protein is a special kind of protein called a transcription factor. Transcription factors are proteins that are unique because they can control the output of protein production from other genes. They bind directly to the DNA of the genes that they control, like a hand turning a light switch on or off, where the switch represents the control of many genes. A normal RUNX1 protein acts by turning on the production of some proteins and turning off the production of other proteins. In the case of a germline RUNX1 mutation that causes the production of an abnormal RUNX1 protein, the abnormal protein often can't bind properly to the DNA of the genes it's supposed to control, which then either reduces the overall protein levels that should have been turned on, or on the flip side, if RUNX1 was supposed to turn off a gene, then that gene's protein level may end up being too high. One can think of the blood system as a family tree. At the very top of the tree is the blood stem cell. She is the mother of all the cells below. We have roughly 100,000 blood stem cells in our body, and they are responsible for making nearly 1 trillion blood cells each day. Researchers are still learning a lot about the role of RUNX1 in stem cells. What we do know is that having low RUNX1 levels can affect how often stem cells divide to make copies of itself, and it can skew the stem cell to make more myeloid cells and simultaneously make fewer lymphoid cells. We also know that RUNX1 is important in the lymphoid cells, your B and T cells specifically, yet we don't understand this as well as we understand RUNX1's role in platelet biology. Having lower RUNX1 protein levels, as is the case in RUNX1 FPD patients, causes lower production in platelet number and can also cause poor functioning platelets, which is why most patients experience easy bruising and prolonged bleeding upon time of injury or surgery. Platelets are central to helping our bodies clot and protect us from bleeding out. We hope to learn more about other cell types within the blood in the near future, like neutrophils, for example. Dr. Nancy Speck and her team have found that in mice with low RUNX1 levels, neutrophils behave differently than in mice with normal RUNX1 levels. They appear to be hyperreactive to infections, releasing high levels of pro-inflammatory proteins. And we know that inflammation plays a role in cancer growth and development. Time will tell whether this is true in humans with low RUNX1 protein levels as well. Studies across many families who have RUNX1 mutations also show that there is a higher number of individuals who develop blood cancers. In fact, estimates suggest that the lifetime risk of developing a blood cancer in RUNX1 FPD individuals is between 35 and 50%. On average, that is 30 times higher than the general population. So why is there this dramatic increased risk? We don't know all the reasons for this, but we do have some clues that have helped us strategize on how best to further our understanding of all the necessary factors that play a role in the evolution of blood cancer, and in parallel, begin to test a variety of potential treatment directions that could delay or block cancer in RUNX1 FPD.